So the mitochondria really has not only this crucial role in energy generation, it has such a central role in cell biology. And because of this, it's really no surprise that the organelle is a dominant site for the initiation and propagation of many disease processes, not just mito. So here in this diagram, you can just see some examples of neurological conditions linked to mitochondrial dysfunction, Parkinson's, migraines, a number of neurological conditions, dementia, and then we have non-neurological conditions like diabetes, pancreatitis, etc. So we are obviously interested in mitochondrial disease for a number of reasons. And these are broadly, as many of you would know, categorized as diseases of energy production or defects in energy production. As a scientist, I was really drawn to these conditions because they are what we call genetically very heterogeneous. So there are greater than 280 genes that are known to cause mitochondrial disease if they are mutated. This is quite extraordinary and it's quite phenomenal actually. And in many ways, as a scientist, it's really intriguing, but in many ways, this is probably one of the reasons why we can't find an effective therapy for mitochondrial diseases, because they are clinically very heterogeneous. So the large number of genes that are associated with these disorders obviously gives rise to many different clinical presentations. And as a consequence of this, development of effective therapies can be challenging. In many ways, this heterogeneity, I think, also makes it very challenging for the broader community to be able to connect with mitochondrial diseases, because it's not like some conditions, for example, like Huntington's disease, which is associated with the Huntington gene, or with breast cancer that's associated with the BRCA genes. People can associate some of those conditions with individual genes and they can connect to it. And I think with mito, that's definitely more challenging because of the large variation in terms of the number of genes that can cause this disease. And this is where it's very important for people like myself to be able to communicate. I guess even though there are many genes that cause these diseases, the overall condition of mitochondrial disease is very prevalent in our community, with 1 in 5,000 live births being affected by mito. So if you think about mitochondrial diseases, what many people think or attribute these diseases to are defects in energy production. So what I've circled here, so this is a very big mitochondria, and in red I've circled these proteins. These are, it's like a little city of proteins that we call the electron transport chain. And the electron transport chain has a very specific function in generating energy in mitochondria. And we know when you mess about with these complexes, it gives rise to mito. Okay, so any perturbation in the in this pathway, or putting these proteins in mitochondria, or putting them together somehow in their building blocks, gives rise to mito. And we refer to this, anything related to the electron transport chain, we refer to this as primary mitochondrial disease. But what's emerging, and definitely what's emerging over time, is that we have this second category of mitochondrial disease, which is called secondary mito. And this is the form of mitochondrial disease that my lab studies. And this form of mito is basically linked to genes that don't directly participate in the electron transport chain. So they may indirectly affect the electron transport chain, or they have some total other role in mitochondria, and hence we call it secondary mito. I hope what you can see in this graph, so in blue is the number of genes linked to primary mitochondrial disease through the years, and you can see it's growing. But over the years, what you can see here in orange is the number of genes linked to secondary mitochondrial disease. And originally, we thought for many years that it was just the electron transport chain that was probably connected to mitochondrial disease. But as DNA sequencing technologies have advanced, and just as our capacity to undertake particular types of experiments have advanced, what you can see is that here in orange, the number of genes linked to secondary mitochondrial disease has basically surpassed the number of genes linked to primary mitochondrial disease. And so I think that this is really a really crucial area of research to start investigating. And it gives us the opportunity to study not only these processes in the context of mitochondrial disease, but also in the context of health and what they do in healthy and functional mitochondria. Okay, so if we now just walk through or take a closer look at secondary mitochondrial disease, I don't want you to be overwhelmed by these terms. I don't expect anyone to know what they are, but we can categorize the genes that are secondary mitochondrial genes into a number of different pathways that take place in mitochondria. Now, I'm specifically interested in this light blue quadrant here, which accounts for 14% of these secondary mitochondrial disorders, and we refer to this process as protein import. So I've drawn this little diagram here to try and describe what protein import is. So like I mentioned earlier, there's about 1,200 proteins that function in the mitochondria, but they're encoded here in the nucleus. Now, these 1,200 proteins are not all produced at once. The cell is very smart. It produces proteins as it needs them. And so in this example, I've shown you here a mitochondrial gene, and it gets converted into a message. And this message can get exported out of this nuclear region, and it finds these protein-producing factories, the ribosomes. And the ribosomes now will produce the protein. This is a mitochondrial protein, but the mitochondria is over here. And the process of delivering this protein, to the mitochondria is referred to as protein import. It's like importing goods into Australia. We need to import these proteins into the mitochondria where they function. And to do this, we have these very specialized machines. I like to refer to them like railways. They provide like paths that allow the proteins from here, this blue one, to find its way into the mitochondria. And it's these particular translocases, these little boxes that I've shown here, that my lab is focusing on. So I'm going to enhance the level of complexity here. Please don't be overwhelmed. But what this diagram is meant to simply depict is our current state of knowledge of these translocases. Okay, so this is what we know. They are composed themselves of different proteins, but every one that has a color to it has been assigned a mitochondrial, has been linked to a mitochondrial disease. So this process of protein import was identified like 20 or 30 years ago. And never would they have imagined that these proteins would be linked to mitochondrial disease at that time. But like I said, as we advance in our capacity to do research, but also in our capacity to sequence the genome, more and more cases are emerging like this, where basic biological processes are linked to diseases like mitochondrial disease.